Okay, so this is episode 5 of the first chapter here, Evolution and Development, in this section. So what we're going to talk about here, a couple of developmental concepts that are somewhat related to the developmental process. And so obviously if you've taken courses like 106 or the maybe vertebrate zoology or even some of Dr. Wrench's plant classes, you'll know that the process by which organisms will develop and their anatomy is very much related to their ancestry and their relatedness to each other. So these processes that you and I will of course be discussing over the duration of this course are very much related to the ancestry of two species. So I guess the easiest way to think about this is if two species have a similar body plan, then you'd expect them to be built by a similar process. So obviously lions and tigers, because they look so similar, it wouldn't surprise you that the way in which they go about being built is going to take place, or is going to take place by similar processes. And if you even go a little bit further out from that, because lions and humans are mammals, you would expect a lot of the processes by which the two of those species are built would be very similar. But what's really actually kind of interesting as well is that even when two things aren't similar necessarily in an evolutionary scale, they oftentimes will still show relationships in that they still are going to use the same molecular tools. So, for example, and I'll show you a picture in a second, the same regulatory protein used to build fruit fly eyes is used to build mammal eyes. So you guys have probably seen what a fly eyeball looks like with its sort of multifaceted receptors and so on. And of course the human eye here is very different from that. And you would sort of imagine that the building of the human eye versus the human building of the Drosophila eye would be very different. And indeed, some of the steps to build these two eyeballs are very, very unique. But what's really unusual or really interesting about this particular story is that there's one protein that has been discovered that is very much used in the building of both eyeballs. And so in humans we call that protein PAC6, and in fruit flies it's called the eyeless protein. And basically what this is a transcription factor. It controls gene expression, whether genes get turned on or off at the same times. And this same protein called eyeless or PAC6 is actually responsible for genes that are going to be needed for building an eyeball, whether it's an eyeball like this in fruit flies or whether it's an eyeball like this in humans. And if you mutate that particular gene in either of the species, you end up or as you might expect, you end up messing up eye development in both. The second example I had here was the presence of the same regulatory protein in limbs is actually going to result in very dramatic differences in two different species as well, like bats and mice. So let's look at that example here. So obviously the layout of a hand, and in this case the what we could call the forelimb, the way in which the bones are laid out and the way in which the fingers are spaced and so on, the mechanisms by which all of that takes place is actually almost identical in both mice and bats. You'll see all the same bones at the same locations, and it's regulatory proteins that are responsible for a couple of things here. One would be basically the stimulation, if you will, of the skeleton. So you'll notice in the bat, these finger bones are very long and extended. So that's caused by regulation. You see the layout is in the same order, but of course the prominence, I guess you would call it, of some of these different digits might be different for some of us. Of course, neither of these have a really prominent thumb, but of course you and I do. And another major difference caused by a couple of regulatory proteins would be, of course, in bats, we've got this webbing between the digits, whereas in the mice that webbing goes away. And so here you can see the development of a limb in something like a mouse. Here would be the development in a bat. So you can see the long extended bones, you can see that webbing, and here you can see where the webbing is starting to go away. So this is controlled by the same kinds of proteins that are basically expressed at different times for different duration, and that's what's responsible for these differences in a couple of different species. The other thing I'd like to highlight here too is what we talked about in the last session, where notice how general develops slowly over time into more specific and more unique characteristics. So notice how they start out the same or very similar, but the older we get or the further along we get in development, the more and more unique we get, so that by the time development's over, we have completely different forelimbs in both the mouse and the bat. Okay, so I think I'm going to end that one there, and we'll pick up with another aspect of evolution and development in the next session.